Good morning, students, and welcome to a new class of 8th Standard Biology. Today we are going to discuss a new topic, and that is compound microscope. So, how do you think we can see very minute objects? We can see very minute objects which is invisible to human eyes through microscope. If we want to see blood cells, some bacteria, virus, which is invisible to our eyes, we use microscopes, and they help to enlarge the object and to view the image larger. Okay, so now we have to check what are the different types of microscopes, and they are simple microscope, compound microscope, and electron microscope. So we will look what is the simple microscope is. A simple microscope is a basic optical instrument that uses a single convex lens to magnify small objects, and it functions as a magnifying glass, and it enlarges the size of the image to 10 times. It's just a basic optical instrument, and it only uses one lens. A single lens is used in a simple microscope. You can see the picture of simple microscope in the slide. Okay, do you understand? Okay, that is the simple microscope. Next is the compound microscope. A compound microscope is a microscope in which more than one lens is used, and it magnifies an object up to 1000 times. Okay, that's the difference between simple and compound microscope. Simple uses only one lens, whereas compound uses more than one lens. Simple magnify object up to 10 times, whereas compound microscope magnify object up to 1000 times. Do you understand? Okay, now we will check what are the different parts of a common microscope, and all these parts have different functions. Okay, the parts are given in the picture. So look at the picture. Okay, in the topmost position there is the eyepiece. Next is the body tube. There is coarse focus knob and fine focus knob, arm, base, revolving nose piece, objectives, stage clip, stage, condenser, iris diaphragm, and mirror. These are the different parts of a compound microscope. Okay, functions of different parts of microscope is written here. First we will look the function of eyepiece. Eyepiece is also known as ocular lens. It is the top lens you look through. Okay, when we take a microscope, we look through what? The eyepiece. It magnifies the image formed by the objective lens, usually up to 10 times. Okay, the objective lens make an image, and the image is magnified by the eyepiece, usually up to 10 times. Okay, next is the body tube. Body tube holds the eyepiece and objective lenses at the correct distance, ensures light travels properly from objective to eyepiece. Okay. Body tube holds the eyepiece and objective lens at the correct distance. Next is the revolving nose piece. Revolving nose piece holds the objective lenses and can be rotated to change magnification. If we want to look through different lenses, we can rotate the objectives by rotating the revolving nose piece. Next is the objective lenses. It is the main magnifying lenses of microscope, and the magnifying power of these lenses are 40x and 100x. I told you we can change the objective lens according to our wish. There will be 3 of them, and the magnification power is given, that is 10x, 40x, and 100x. It produces the first magnified image of the specimen. Okay, the objective lens magnifies the image of the specimen. Next is the stage. Flat platform where the slide is placed for viewing is the stage. Okay, the slide is placed on the stage for viewing. The specimen is placed on the slide, and the slide is placed on the stage. Okay, we look through the specimen when it is kept on the stage. Next is the stage clip holds the slide on the stage. Okay, if there is no stage clip present, the slide could move from the given position. So if you want to hold it tightly, we can use stage clips. Next is the condenser. It focuses light onto the specimen. It helps produce a clearer, brighter image. Next is the iris diaphragm. It controls the amount of light passing through the condenser. It adjusts brightness and contrast. Okay, there will be a lot of light passing through coming from the mirror, and the iris diaphragm has to adjust the amount of light passing through the condenser. It can be in a large amount or a small amount, but the iris diaphragm helps to control the amount of light falling on the condenser. Next is the mirror. It directs light upward through the diaphragm, condenser and specimen. Okay, the mirror is placed on the down portion of the microscope and it directs the light upwards through diaphragm. If there is no enough light present, we cannot view the specimen properly. Next is the coarse focus knob or coarse adjustment knob. It moves the body tube up and down quickly. For precise viewing of the specimen, we can move the coarse focus knob, and it is used for initial rough focusing under low power. Next is the fine focus knob or fine adjustment knob. It helps the move the stage slightly for precise sharp focusing, especially under high power. Okay, fine focus knob is used to move the stage slightly for precise focusing. Okay, next is the arm of the microscope. It supports the body tube, nosepiece and stage. It is used for carrying microscope safely. If you want to carry a microscope from one place to another, we have to hold on the arm of the microscope. Next is the base. Base supports the microscope, holds the entire instrument steady. Do you understand? Okay, base supports the microscope and holds the instrument steady. Okay, now we have to check what is magnification power. Magnification power of a lens is its ability to magnify objects. The magnification power is the ability of a lens to magnify the object. How much a lens can magnify object is called magnifying power. If the eyepiece lens magnifies the object to 10x and the objective lens magnifies the object into 40x, then the total magnification power will be 400x. Okay, that is the. Okay, next is the arm. Okay, today we discussed about types of microscopes, simple microscope and common microscope. Then we discussed the functions and different parts of microscopes. And okay, you have to go through the class clearly and thoroughly. The eyepiece and objective lens are the important of among these. You have to study it properly. Okay, thank you for watching.